News release. Monday, May 8, 2023. Discovery provides potential clues for Lewy body and frontotemporal dementias. Scientists at the National Institutes of Health have identified new genetic risk factors for two types of non-Alzheimer's dementia. These findings were published in Cell Genomics and detail how researchers identified large-scale DNA changes, known as structural variants, by analyzing thousands of DNA samples. The team discovered several structural variants that could be risk factors: Lewy body dementia L, B, D, and frontotemporal dementia F, T, D. The project was a collaborative effort between scientists at the National Institute of Neurological Disorders and Stroke NINDS and the National Institute on Aging NI at National Institute of Health. Structural variants have been implicated in a variety of neurological disorders. Unlike more commonly studied mutations, which often affect one or a few DNA building blocks called nucleotides, structural variants represent at least 50, but often hundreds or even thousands of nucleotides at once, making them more challenging to study. If you imagine that our entire genetic code is a book, a structural variant would be a paragraph, page, or even an entire chapter that has been removed, duplicated, or inserted in the wrong place, said Sonia W. Scholes, MD, Ph.D. investigator in the neurogenetics branch of NINDS and senior author of this study. By combining cutting-edge computer algorithms capable of mapping structural variations across the whole genome with machine learning, the research team analyzed whole genome data from thousands of patient samples and several thousand unaffected controls. A previously unknown variant in the gene TCP9 was found in samples from patients with LBD, a disease that, like Parkinson's disease, is associated with abnormal deposits of the protein alpha-synuclein in the brain. This variant, in which more than 300 nucleotides are deleted from the gene, is associated with a higher risk for developing LBD. While this finding is new for LBD, TCP9 is a known risk factor for Alzheimer's disease, which could mean that this structural variant plays a role in the broader dementia population. From a genetics standpoint, this is a very exciting finding, said Dr. Scholes. It provides a point of reference for cell biology and animal model studies and possibly down the road a target for intervention. By looking at a group of 50 genes implicated in inherited neurodegenerative diseases, the investigators were able to identify additional rare structural variants, including several that are known to cause disease. The analyses also identified two well-established risk factors for FTD changes in the C9orf72 and mapped genes. These proof-of-concept findings bolstered the strength of the study's new findings by demonstrating that the algorithms were properly working. Because reference maps for currently available structural variants are limited, the researchers generated a catalog based on the data obtained in these analyses. The analysis code and all the raw data are now available to the scientific community for use in their studies. An interactive app also allows investigators to study their genes of interest and ask which variants are present in controls vs. L, B, G, or F, T, D cases. The authors assert these resources may make complex genetic data more accessible to non-bioinformatics experts, which will accelerate the pace of discovery. Research to unravel the intricate genetic architecture of neurodegenerative diseases is resulting in significant advances in scientific understanding, said Brian J. Traynor, MD, Ph.D., senior investigator at NIH. With each discovery, we shed light on the mechanisms behind neuronal cell death or dysfunction, paving the way for precision medicine to combat these debilitating and fatal disorders. Researchers expect that the dataset will continue to grow as additional data are analyzed. This work was supported in part by the Intramural Research Program at NIH I, N, D, S, and N, I, A, N, I, N, D, S, https://www.nins. NAGOV is the nation's leading funder of research on the brain and nervous system. The mission of NINDS is to seek fundamental knowledge about the brain and nervous system and to use that knowledge to reduce the burden of neurological disease. About the National Institute on Aging, NIANI leads the U.S. federal government effort to conduct and support research on aging and the health and well-being of older people. Learn more about age-related cognitive change and neurodegenerative diseases via NIA's Alzheimer's and related dementias education and referral our center website. Visit the main NIA website for information about a range of aging topics in English and Spanish and stay connected. About the National Institutes of Health, National Institute of Health, National Institute of Health, the nation's medical research agency, includes 27 institutes and centers and is a component of the U.S. Department of Health and Human Services. National Institute of Health is the primary federal agency conducting and supporting basic, clinical, and translational medical research and is investigating the causes, treatments, and cures for both common and rare diseases. For more information about National Institute of Health and its programs, visit www.nagov. National Institute of Health Turning Discovery into Health Registered. Pavlovic et al. Genome-wide structural variant analysis identifies risk loci for non-Alzheimer's dementias. Cell Genomics, May 4, 2023. TOI 10.1016/JX Gen 2023.100,316.